Okay, so I'll tell you about our work on immune receptors in plants and plant pathology. And maybe as a way of introduction, I should highlight that next year will be the International Year of Plant Health. That's what the United Nations has decided. And this is because we lose a lot of food to plant pathogens. And there are serious problems like this is wheat blast in Southeast Asia, primarily Bangladesh now. So there's many problems like that that need to be addressed and are really threat to food production and food security. Yet, despite all of that, plants are actually very good at fighting off pathogens. And one of the dogma in plant pathology, I guess, is that most plants are resistant to most pathogens. And sort of illustrated here. So the disease is really the exception. When you walk around here on this beautiful campus, you notice that most plants are actually doing fine, even though they're bombarded by pathogens all the time. And the reason for that, and I'm sure many people on this campus who are primarily biomedical people don't truly appreciate that, is that plants have an immune system, a very effective immune system. And this is really what came out of a lot of research in the last, I would say, 20 years, is that plants are resistant to most pathogens because they're actively resistant. They have an active immune system that allows them to fight off these pathogens. But when pathogens break those barriers and can infect the plant, they often do so thanks to these effectors that were already introduced a few times at this meeting, notably by Bart a little earlier. And so these pathogens can secrete effectors, and there's a variety of different pathogens do that, pretty much all of them. And some of these effectors are functioning inside the plant cell, others are functioning in the apoplastic space. And these effectors will act on targets, either inhibit them, activate them, or bring together different pathways and connect them, essentially create a short circuit in the plant. And so by altering plant processes in these various ways, these effectors essentially help the microbe colonize the plant. And they often, not always, but they often suppress immunity, and that's a primary function of effectors. But other effectors do other things related to nutrition, spread, and so on. But plants don't sit idle, and plants have a very effective set of immune receptors that allow them to detect these pathogens, and in particular the pathogen effectors. And there's two major classes of immune receptors in plants. There's the cell surface ones, which we call pattern recognition receptors, because they often, not always again, but they often detect conserved molecular patterns like bacterial flagellin would be a classic example. And then there's the intracellular immune receptors, which belong to this class, the NLR or MBSLRs, depending on where you read the literature. These are modular proteins that are detecting the intracellular effectors, the ones that are delivered inside the plant cell. They can detect these effectors in various ways, either by directly binding them or indirectly by detecting the perturbations that these effectors would cause. And all it takes really is for a plant to have one of these immune receptors that can detect one of these effectors for the plant to win that interaction and to essentially mount a very effective immune response that will block the pathogen. And you can already imagine that this creates a very unstable evolutionary situation because these effectors that are evolving to essentially favor the pathogens are in these conditions, when the pathogen lands on a plant that has an immune receptor that's matching one of those effectors, they become a liability. They trip the wire, as it says on this slide, and they are now triggering responses that are negative for the pathogen, essentially altering the fitness of that pathogen. It's essentially game over for the pathogen when there is an immune receptor. So traditionally, these immune receptors were defined initially genetically as segregating genes in populations that vary in their resistance levels. And these genes tend to be called R genes. And these resistance genes, this actually leads me to our lunch discussion, the majority of them, but not all of them, the majority of them turn out to code for these immune receptors. So this is really dominant form of immunity in plants. Okay, so a few slides I added this morning thinking about these systems and really to illustrate maybe a theme that came up a few times at this meeting is This cartoon really is best illustrated like that, because for every interaction you take, there is really huge diversity going on. And it's well established now that both on the pathogen side and on the plant side, these effectors and the immune receptors are the most diverse genes in these species. And so there's very active coevolutionary dynamics here that is really resulting in a lot of diversification of both of these players in these interactions. So really, the way to think about would be really a lot of diverse interactions going on within one single system. And of course, there are lots of systems in these plant pathogen interactions. But one of the consequences of these coevolutionary dances that are going on is that the pathogens, because they're rapidly evolving, because they continuously have to keep up with the plant immune systems, they end up shaping this immune system, and ultimately they essentially drive the evolution of the plant genome. And this is something I will get back to at the end, and you'll see how the pathogens have actually shaped the evolution of the plant genome by essentially triggering the diversification of these plant immune receptor genes. But another example I thought I'll include here. I feel very special to give a talk on this campus all the time, because this is primarily biomedical campus, and I think also it came up in the discussion last night with the public event how how this plant blindness, that's really happening a lot with our biomedical colleagues. They don't realize the links that our science has to theirs, and plant science is underfunded and all that. So I added this slide really just if it happens that there's some people from the biomed world who somehow accidentally managed to make their way to this conference, which I really doubt, then this slide is for you. The fact is that these NLR genes, these immune receptors, they occur not just in plants. They occur in fungi, amazingly enough, and they also occur in animals, in mammals. So we have immune receptors. And those people who suffer, for example, from Crohn's disease, these are actually autoimmune mutations in NLR receptors. And also something that's not well appreciated, maybe not widely known, is when the first human NLR gene was cloned. That was a Crohn's disease gene. It's a gene called NOD1. And when that gene was cloned around 2000, that happened to be about 5-6 years after the first plant NLRs were discovered. In fact, that paper recited the plant paper. So there is a link between human immunity and plant innate immunity in the sense that these NLRs are actually probably ancient genes that have diversified on these different taxons. But one interesting differences between mammalian NLRs and plant NLRs is that typically mammalian NLRs tend to be quite conserved across long evolutionary periods. So the split between human and mouse is about 90 million years, give and take. But if you look at the NLR genes, typically you would be able to easily identify orthologs between human and mouse NLRs. And that's not the case in plants. If you compare tomato and coffee, for example, the great majority of the NLRs, not all of them, there are a few that are conserved, but the majority of them tend to have hugely diversified with a lot of birth and death evolution going on over the same period of time. And tomato coffee, by the way, it's about the same split as human mouse, about 90 million years or so. The reason is really interesting, and I think drives a point I again want to highlight, is the reason is straightforward, because these guys are co-evolving with the effectors. They're co-evolving with these rapidly evolving, rapidly diversifying pathogen effectors. So to keep up with that, the plant has to really continuously innovate in terms of its NLRs. In contrast, in mammals, we have an adaptive immune system, and that's what usually takes care of the rapidly evolving pathogens, and that's also adaptive, so it depends on what you get infected with. The immune system will adapt to that. But these NLRs are actually involved in detecting the molecular patterns. For example, mammalian NLRs detect flagellin, which is a functioning plant done by the cell surface receptors. And because these molecular patterns don't change a lot over time, bacterial flagellin is probably about the same over these 100 million years, then there is no really selection pressure on these NLRs to diversify in the same way as the plant NLRs diversify. Okay, so that was my introduction. I want to now give you a little bit of historical perspective on on the resistance. I think it's a good starting point, really, to my talk and to the theme I want to develop. So over a century ago, Roland Biffen, a Brit who was actually working at the predecessor of the John Innes Center, was making crosses between different wheat varieties that had very different resistance levels to rust fungi. And that was soon after Mendel's laws were discovered, and he noticed that there are major resistance genes, as he called them, that were segregating. So single genes, often dominant, were segregating in these populations, and these were defining the resistance levels. And he essentially discovered really our genes essentially with that work. And he was a Brit with a sense of humor, as you can see there. That's not a normal wheat spike. So fast forward 50 years later to Harold Floor. Harold Floor in the US, he brought the pathogen to the equation, and he came up with the famous gene-for-gene model in which on the pathogen side, there is also segregation of single genetic factors. He called them AVRs. We now call them effectors. But basically, you need an effector to match an R gene to have resistance. So that's basically Floor's contribution. And if either one of these factors is missing, then you don't have the resistance response. You don't have the immune response, and you get disease. So Floor's model was extremely influential and really useful in guiding applied research, guiding plant breeding, in understanding also what happens on the pathogen side when pathogens break the resistance. But also it was very fundamental for guiding the basic research, because a major goal in plant pathology was, molecular plant pathology, was to clone actually these components. And that started happening in the 1980s with the first effectors being cloned, and then the first R genes came up soon after that. And then we started having essentially an idea about what's going on at the molecular level. But the point also I want to make is that these models are really great, but as we know, and as we heard also at this meeting, is this concept of single genes are a little bit oversimplified. And when we say there's a gene for resistance, there's a gene for virulence, there's a gene for this or gene for that, clearly that's a simplification. We always appreciate that there is a more complex genetic architecture underpinning these traits. It just happens that those genes are the ones that are variable in the populations we study, right? So we depend, in these classical genetic approaches, we depend on natural variation to identify the components. If those components are not variable, doesn't mean they're not involved in that trait, they're not involved in that phenotype. Of course they are, but they just will be, the geneticist will be blind to those traits because they will not be variable. And in addition to that, there's other factors that complicate genetic analysis. I don't think they're appreciated as much, to be honest, but I think they're factors that are extremely important. Redundancy, obviously, that confounds genetic analysis. Epistasis, and also quantitative traits with complicated genetic architecture underpinning them, that also can make genetic analysis more difficult. So the message, again, I have also for this talk is that we are now maybe in a position to start thinking beyond single genes and start thinking more in terms of systems. And if you were to think about the pathogen-plant interactions, this is probably more closer to the reality than Floor's gene-for-gene model. If you think of the effectors as single red dots, and then the plant factors or plant receptors as blue dots, this is closer to the reality than Floor's gene-for-gene model, which is still true, because many of these interactions at the basis are gene-for-gene, but still maybe we are in a position now to go beyond that model. Okay, so then by the 1990s, the first immune receptors were discovered. There was also an ever-expanding list of effectors with also a lot of variants of these effectors that break the immunity, and that's actually something very unique to plant pathology. We heard also yesterday how it's difficult in evolutionary biology to identify adaptive mutations, but plant pathologists have tons of that because these pathogens often evolve races that are virulent on these immune plants, and those have mutations in the effectors. This can be loss of function mutations, but in other cases, this can also be single amino acid changes. And those mutations, we have very, very long catalogs. We literally have dozens and dozens of those examples. It's just really beautiful. And that having that genetic identity of these various players really allowed us to build this biochemical model of the gene for gene, which is really a receptor ligand type model. So it became very clear that probably what's going on is a receptor on the plant side detecting a ligand from the pathogen, and there's some signaling pathway leading to some immune response, right? So this model was really dominating the thinking for quite a while, especially after the 90s when the first immune receptors were discovered. But again, the reality turned out to be more complex, and this is what we think today, and we think that this is probably reflecting, again, reality much more so than the simpler model. And there's complexity at different levels. So for example, the detection of the effectors by the immune receptors is not often direct. It often involves other factors. And it makes a lot of sense, actually, for the plant to detect these effectors indirectly, because then it can, if you like, guard some important processes that are often targeted by pathogens. And when those processes are perturbed, then the plant can activate immune response. Essentially, if you want to find if you have a mouse or not in your house, in your kitchen, very often you don't see the mouse, but you find a hole in your cereal box. And that's basically telltale that you have a rodent of some type. It may not be mouse, might be rat or something, but that basically means you should call pest control. So it's a bit like that. So that's kind of the metaphor. Direct detection can happen, but it's often not the case. And then we have complexity at other levels where it turned out that these immune receptors themselves form networks. And this is, of course, some people think, oh, yeah, a network. Yeah, of course, every signaling pathway is a network. Yes, indeed, every signaling pathway is a network. Every biological process is a network. But in this case, it's the immune receptors themselves that are forming networks. So they have diversified into working with each other to essentially produce a potent immune response. And those networks can be complicated. For example, there can be redundancy. Like this guy here is working with 1, 2, 3 components that are downstream or co-receptors or helpers, whatever you want to call them. So this is really another, again, layer of complexity in these networks. But the one feature that is very apparent, and that's true both for the cell surface receptors and for the intracellular NLRs, is this uncoupling of the sensing of the pathogen from the signaling itself. So this is a very clear feature of all these immune receptors. And this is actually, by the way, parallels what happens in mammalian immunity, where these receptors have now evolved to have these different functions, where a subset of them are involved in sensing the pathogens, and others are involved in delivering the immune response. And that has a lot of implications. And this is summarized in this slide here. Why do we have an immune receptor network? Why do we have a redundant network? And one of the features is probably that when you uncouple recognition from signaling, you create a system that's a lot more evolvable than if those two functions were constrained in the same protein. So by having this uncoupling, you essentially increase the adaptive landscape, the number of mutations that could arise that will allow the plant to keep up with the pathogen is probably wider. And so that's probably more effective immune system. But another feature of network is robustness, and here we have redundancy. Same reason why we have two engines in an airplane. If one of them breaks down, then the airplane can still fly, it doesn't crash. Same thing here, if you have redundancy of these core elements, then it's more likely a more stable and more resilient system, especially when that system can be targeted by pathogens for suppression. So if you have redundancy at that level, then when a pathogen evolves to suppress the system, then your system will still function because you have redundancy. Okay, so that's kind of a little bit the second introduction, if you like, and it gives a little bit the theoretical framework for what I want to talk about. Let's now move to the system itself. So the system we study is these NLR intracellular receptors. These are multi-domain sensors. In plants, they have this variable domain, so they have a very conserved nucleotide binding domain in the middle, loose and rich repeat at the C-terminus, and in plants there are 3 different types of N-terminal domains, the coiled coil, a variant on the coiled coil, and the tear domain. And I will talk today about the coiled coil type NLRs. And the readout in plant biology is when these NLRs are activated, you get a cell death response, the so-called hypersensitive cell death. You can see it if you put spores of a pathogen. This is the potato blight pathogen on leaf. This guy here is detected by the immune receptor. This one is not. And you can see this patch of dead cells. That's the output of the immune response. And we can recreate this experimentally by expressing the pairs, expressing an immune receptor with its matching effector in an experimental system like Nicotiana benthamiana. Then we can see that cell death response only when the two partners are present and not in the other cases. This is essentially recreating Floor's gene-for-gene system experimentally in benthamiana leaf. So for a long time, these genes were first cloned again in the early 90s. For a long time, there was a lot of work chasing the downstream elements. And that had yielded essentially very limited success. It turned out to be really difficult to identify, in particular, classic signaling pathway underpinning these immune receptors. And only recently we know why this is the case, because it turned out that these NLRs function through this type of machinery where when an inactive NLR, so these are kept in an inactive state as ADP-bound state, when those NLRs are activated by the pathogen effector, this triggers essentially the release of the ADP binding to ATP, and it triggers this oligomerization. This is what our colleagues in Beijing, Jiamin Zhu and Jiji Shai, called the resistosome. They published this only recently, last April. And this resistosome is, in this case, is a pentamer. This is an NLR called ZAR1. It's from Arabidopsis, detects a bacterial pathogen. And that oligomerization process, essentially this molecular wheel that is formed, is also associated with the flip of the N-terminal alpha helix. So the absolute N-terminus there, shown in red, flips out and forms this funnel-like structure that is probably perturbing the membrane in some ways. This part is less clear, but it's thought to cause a cell death directly by inserting into the membrane, possibly through pore-forming activity, or maybe by acting as an anion channel, maybe calcium channel. This results in the cell death response. So this is not your classic signaling pathway, but this is a more direct way of causing the cell death response that is caused by the activation of this molecular machine, the resistosome. And I'll get back to this later. This will be relevant later on. So the network we work on is the so-called NRC network. This is called the NRC network because the downstream players are what we call NRC, NLRs required for resistance, for cell death. And this network is quite a complex one because it's involved in detecting a lot of different pathogens, anywhere from nematodes, aphids, bacteria, viruses, and oomycetes, or Phytophthora, the potato blight. And we discovered this network not that long ago. In fact, the paper was published only in 2017, but this was work from my PhD student who was interested in this gene called RPI-BILB2. So my background, actually, I come to this field from the pathogen side. So we discovered a lot of effectors. Some of them activate immunity. One of these effectors is called AVR-BILB2. It comes from the potato blight pathogen. So we're interested in studying that. It activates an NLR called RPI-BILB2. And Shihang Wu, when he was starting his PhD, said, I'm interested in understanding how this works. So he started studying that, and he discovered this NRC4 as a downstream element. And that's an NLR that is kind of related to RPI-BILB2. But that was a strike of luck, really, because as you can see here, RPI-BILB2 is only dependent on NRC4, is not redundantly using other NRCs. For example, RX, discovered by David Wellcome, virus resistance, works with NRC2, NRC3, and NRC4. Same with BS2, Brian Staskovic, bacterial spot, NRC2, 3, and 4. So those guys couldn't be identified readily by genetic analysis when you have this type of redundancy. So we were fortunate to discover that, and then through phylogenetic detective work, Shihan could unravel this entire network by basically combining phylogenetic analysis with functional data. But this turned out to be a really classic NLR network. But perhaps more interestingly than a network is the evolution of this network. And when we map the position of these NLRs and this network on a big phylogenetic tree of NLRs, so this is covering a lot of diversity of plants, what we notice is that the NRCs, so the downstream component, and all of their sensors form sister clades. This is very well supported clade that has evolved probably about 100 million years ago, actually evolved earlier than 100 million years ago, but diversified about 100 million years ago. So that's where we have these expansions, especially in the sensors, which kind of makes sense, because those are the guys co-evolving with the pathogens. And the downstream elements, they tend to be conserved across plant species, not as diverse, which also explains why a lot of these sensors can be moved around from one species to another. But what you can immediately realize by looking at this network is clearly this network has evolved from a pair of NLRs, and then ultimately before that from a singleton. So this phylogenetic structure suggests that. And the data also does suggest that. So when we look at, I'm going to skip this for the sake of time. When we look at the evolution of this guy, if we look at some of the early species that have this network, like the Caryophyllales, like sugar beet, then we don't find a network. We find essentially what is most likely the ancestral configuration of this network, which is a very simple cluster with one NRC linked to two sensor type NLRs. So this is what happened early in evolution. This was probably singleton that duplicated into this small cluster. But then over 100 million years of evolution, this is what happened in tomato, which is one of the species that has the expanded network. Then these guys went and now basically spread throughout the genome. And you can see there's NRCs scattered throughout the genome and a whole bunch of sensors, often in genetic clusters, as you can see there. So it went from this simple configuration of an NLR pair into this complex and linked genetic network. Okay, so all of this work led us to this model where we now suggest that these NLRs can function as singleton. In fact, the ZAR1 system, the one where the resistosome structure was discovered or unraveled, is a singleton. And those singletons at some point duplicated into sensors and helpers. And that's the consequence of subfunctionalization or specialization, because these singletons can do both functions. And then over evolutionary time, we have this diversification into this network. So the next question. So we're pretty confident that this is the model that takes place, at least in our network. And certainly there's many other examples in the literature of at least of pairs or at least clustered mini networks, if you like. But the question I was really interested in, and this goes sort of straight at one of my main goals for my lab, is really linking a mechanistic understanding of these systems with evolution. And so the question here is, when these evolutionary transitions take place, what are the molecular consequences of these transitions? How does this affect these NLR proteins? How does it affect their functioning? How does it affect their biochemistry? So I'll finish the talk with this story related to this particular question. And to start that particular part is, I started really with the frustration because when Aki joined the lab about 3 years ago, at that time we had a bit of frustration with the NRCs because in many cases with these NLR proteins, if you express the N-terminus by itself, you get autoactivity, you have autoimmunity. And that's probably because the signaling bit is carried by that N-terminus. In the case of the NRCs, we couldn't see that. And we just didn't really understand why it was annoying us because we assumed that's the executor of the cell death. And we reasoned that maybe one reason that didn't work is because we didn't have the right boundary. These domains, at that time at least, were not always clearly defined. And we thought, okay, maybe we had the wrong boundary. And we had also other data suggesting that that could be an issue. So when Aki joined, he decided to have as a side project this transposon mutant screen, where essentially we use a mu transposon, mu, and in vitro mutate the NLR. This mu will introduce stop codons, so essentially you make truncation library of your NLR, and then express it in plant and look for autoimmunity. So he did that with one of the NRCs, with NRC4. And essentially, it kind of didn't work because, as you can see here, the majority of these truncations, we had, I think, 65 in this particular case, are blue, which means there's no cell death, no activity. However, he did get one of these guys to be autoactive, and that's that little guy there, which has the first 29 amino acids of this protein. So the absolute N-terminus of this NLR was sufficient to give autoactivity. So we looked at each other actually with some puzzlement. We didn't really know how to interpret this. We noticed that this sequence was fairly conserved across many NLRs. But to be honest with you, we kind of ignored it for a while. We did confirm it, though. We took the 29 amino acids and a series of experiments. Don't want to get into. The bottom line is you need to fuse it to YFP to have the activity, to have the cell death activity in a robust way. The absolute N-terminus of NRC4 fused to YFP gives you a cell death, and it's not dependent on the full-length NLR. So if you take an NLR knockout, these are CRISPR mutants, then you still get that N-terminus to cause cell death. So this is really, by itself, when it's fused to YFP, is sufficient to cause cell death. Further experiments I'll have time to show is that the reason why YFP is required because it triggers oligomerization. YFP is known to oligomerize, and when you fuse that N-terminus to YFP, you make even better oligomers, and that's why you get this activity. We know it's dependent on oligomerization by YFP. Okay, so that was, again, an interesting result that we didn't really know what to make of for a couple of years until the work of Gigi and Jamin came out in last April, and that was really striking to us because the activation of the ZAR1 resistosome is associated with this really dramatic conformational switch where the alpha helix, the N-terminal alpha helix, which is normally buried in the coiled-coiled domain in the inactive form of the NLR, when this guy is activated through the effector and through binding ATP and it oligomerizes, that alpha helix flips out. There's really an absolute dramatic fault switch, which now we refer to as the death switch, actually. And that fault results in that funnel, which is presumably inserting in the membrane and causing some membrane perturbation leading to the cell death. So, of course, when that came up, it was for us a real aha moment, and especially that when we align our NRCs to ZAR1, so the resistosome comes from ZAR1 and our Arabidopsis protein, and if you align that to NRC4, the one we mutagenized, then you can see that the N-terminus is quite conserved. In fact, in that alpha helix, about roughly, the alpha helix is the first 21 amino acids, and roughly half of the residues are invariant between ZAR1 and NRC4. So that turned out to be a really cool finding. We went on and stepped back and did some computational analysis. A technical point here, when you do phylogenies of NLRs, you have to use the MBARC domain because that's the only domain that's conserved across a lot of NLRs. If you use the CC domain, you can't really build phylogeny because it's too diversified. Some of them, you can't even align them. So what we had to do initially is to group them based on sequence similarity. So we used this tribing exercise. And this is how the CC or the N-terminal domains of all these NLRs group. They group in tribes of different sizes. But tribe 2 in particular is the one I want to highlight because tribe 2 includes the NRCs and also ZAR1. So that's primarily a dicot plant tribe. So indeed, the CC domain of ZAR1 is related to the NRCs, and as shown by this analysis. We then looked for motif. We used classic methods, MIME, to find motifs in an unbiased way. And then the tribe 2 had a motif at the N-terminus, which is shown here. And that matches the region we identified as having the autoactivity and also matches that first alpha helix. And this motif, which we called MADA for MADA, that's the beginning of this consensus, is present about 70% of these tribe 2 NLR proteins. And that matches, again, the N-terminus of both NRC4 and ZAR1. So that's the MADA motif. That's where it's located. And what's interesting about it is really actually surprising in a way, but hold on, I'll tell you the answer to that, is that this MADA sequence is found in about 20% of all the CC-NLRs, always at the N-terminus. But it's present in NLRs that are quite divergent from each other, that are not necessarily closely related. So ZAR1 again is from Arabidopsis. The NRCs are here. It went away. They're from Solanaceae plants. That's quite distant. Even rice, for example, this is a monocote. Some of the rice NLRs have also this MADA motif at their N-termini. So it's a sequence that is present in 1/5 of the NLRs, but has been conserved throughout evolution in fairly distant NLR from distant taxa of plants. Okay, so next we had to do functional analysis, of course, to test the functionality of this MADA sequence. And the first thing we did is we recapitulated in our NRCs the mutations that were identified in the ZAR1 study as being essential for the activity. And these are, again, in the N-terminus. This triple mutant was shown in the ZAR1 paper as being required for ZAR1 activity. If we introduce it into our MADA motif of the NRC4, we also lose the activity. So that's, you can see that here, that these mutants, especially if you make the glutamate mutation, which is more disruptive than alanine 1, it abolishes the activity, but it doesn't affect the stability of the protein. We also did a mutational scan of the entire sequence. We did alanine, but that's not very disruptive because it's quite a hydrophobic region. But with glutamate, we could identify 3 independent single mutations that were abolishing the activity of the NRC4. And again, these are in this MADA motif. All of these positions are on the outside of the funnel-like structure. So you can see that that's all on the outer face. And so this is possibly involved in either membrane integration or interaction with other components that are maybe carrying it to the membrane. But we did not identify any mutations in the inner face of this funnel-like structure. Another experiment that was quite evident is to do some domain swapping. So we essentially swapped the N-terminus of NRC4 with the N-terminus of ZAR1 and various other MADA-type NLRs, including some from rice, which are fairly, again, distant. And in every case, we could maintain the functionality of the NRC4 after doing this kind of swaps. And we also want to look beyond the hypersensitive cell death readout. We wanted to look at resistance. So we did also an experiment with the pathogen, with the potato blight pathogen. And this is maybe an interesting one. If you make a ZAR1 and NRC4 chimera, again, with swapping only the alpha helix, then that still is functionally conferring resistance to this pathogen. It's as functional as the wild-type sequence. OK, so back to the computational analysis. You're probably wondering what happened to the sensors NLRs, the ones that are in this NRC network. Do they have this MADA sequence or not? Well, interestingly enough, they have quite different CC domains. And as I told you, the NRCs come in tribe 2 when you group these proteins based on the N-terminal domain. But look at all the sensors. They're all shown in blue here. They're essentially scattered throughout a lot of different tribes. And that's because they have very divergent CC sequences. They don't have the MADA sequence. And their CC domain typically have diversified to the point that they're not really clustering anymore with the NRCs, even though they share a common evolutionary origin, because this is, again, the tree based on the NB-ARC domain, based on the conserved feature. And this is the NRC superclade, which is here. And you can see that all the NRCs, the majority of them, there's a couple of exceptions, have the MADA sequence. But none of these sensors have the MADA sequence. So all of them just lack it. And in fact, this clade here happened to have even an insertion of a domain before the CC domain. So this is an N-terminal extension of about 500 amino acids or so that is involved in detecting and baiting the pathogen effector. So clearly, these guys with that insertion cannot function through this resistosome type model because that requires a free N-terminus to really form that funnel-like structure and function. So what happened? Well, what happened is a typical feature of evolution. Use it or lose it. These guys are now not functioning as executors of the cell death. They depend on the NRCs to do that. So they don't need to maintain a functional CC domain in terms of the resistosome function. So in that case, they can diversify, and they're under relaxed selection as far as that CC domain goes. And in fact, some of them even have acquired these N-terminal extensions, really then diversified even in terms of their modular structure compared to the ancestral state, which was probably an NRCs R1-like type NLR. So this leads us to, again, this model, but now we have some understanding of what happened as these NLRs went from a singleton, again, like the ZAR1 case, and specialized into sensors and helpers, and then even diversified into networks. We think that the MADA type sequence, that alpha helix, has been retained in about 1/5 of these NLRs. So it's been retained both in terms of the conservation of the sequence, but also in terms of the position. So probably all of these guys function through a resistosome type mechanism. But the sensor NLRs, they diversified quite a lot. So their MADA motif typically degenerated. And also in some cases, they acquired new integrations, new domains that are now functioning in detecting the pathogen. And they interact with these NRCs to execute the cell death. So that's a story. This is really a wrap-up of what I told you. I think what's really interesting here is that this sort of evolutionary trajectory where you have a system that originated as a single protein carrying multiple function that has diversified into this basically functionally specialized players, which together form probably a more efficient system. For example, this integration of the N-terminal domain probably wouldn't be able to happen if that protein has to execute the cell death at the same time, because it won't be able to have that free N-terminus. So this really illustrates how a system like that is more efficient into keeping up with the pathogen. And one little perspective here that also highlights something we're really interested in doing at the moment in the lab is one of the things that the MADA predictions allow us to do is it allows us to classify these NLRs into different functional categories. When we sequence plant genomes, essentially we have several hundred, usually NLR genes identified. This is a bit like an alphabet soup, so it's very disorganized. We really don't know how to make sense of that. But the MADA predictions at least allows us to have a first shot at trying to organize these NLRs into functional categories and maybe identify additional networks in these various NLRs. So that's what I had. I want to thank the folks in the lab who did the work. So Shihan is the one who discovered the network in the first place and has been quite an influential force in the lab. He's about to move to start his own lab in Academia Sinica in Taipei. Lida works on effectors that suppress the network. I didn't talk about that today, but we identified also effectors that suppress the NRCs. So they're quite effective effectors because by suppressing NRCs, you really take out of action a big chunk of the NLRs. Abbas does the biochemistry, and also the structural biology, something we're pursuing very actively. Aki is the one who identified the MADA sequence and exploited that. And then other people like Rong, Mauricio, and Toshi have been helping with various aspects of the project. And Mauricio is also working on effectors that suppress the network, and Toshi is involved in network predictions. Also want to thank Toga and his lab. Toga is at Imperial College. He studies the cell biology of these NRCs, and there's some other stories that maybe he'll tell you one day. And then my longtime collaborators, Mark Benfield and Ryohei Terauchi, and NLR collaborators. And also, the other part of my lab works on the blast fungus. I also want to thank them, although I didn't talk about their work. And also I want to thank my colleagues at the Sainsbury lab. So thank you for your attention.
Thanks a lot, Sophien. We have time for questions.
Hi. Yeah, this is really intriguing, and I'm wondering how much flexibility does the network have built in? Like, could multiple helper NLRs form a complex together, or can it be just one? And how would sensor NLRs sort of signal to the helpers to start forming the resistosome?
Yeah, very good question. So. I didn't have time to address that. So the first part of the question is really the maybe related to redundancy also, and how much flexibility does the system have. The one thing, I didn't want to include all the biochemistry here, but the one thing I want to highlight is that we know now, even biochemically, that these guys are really independent pathways. They're truly redundant. So even biochemically, for example, the NRCs form complexes with themselves, but they don't make heterocomplexes. So in the plant cell, there is an NRC2, probably, I mean, we don't know if it's a resistosome or not, but there's an NRC2 complex, and an NRC3 complex, and an NRC4 complex. So probably soon after this diversified, they probably also evolved to be really independent from each other. And that, I think, is truly a redundant system. It's not, many of these network type of architectures are actually built to compute essentially the ideal response depending on different environmental signals, right? But we think this is a truly redundant system. And also the genetics support that. So we actually did experiments with the virus. Rx gives viral resistance. And if we have knock out the 3 NRCs, we can equally complement with either an NRC2, 3, or 4. Each one of them will be equally competent into bringing the resistance level against the virus to its full extent. So that's kind of your first question. The second one was, what was the second one?
How these sensors might
Oh, yeah, right. Yeah, exactly. Yeah, good point. So for that, let me go back to the resistosome model. And where is it? It's here, right? Yeah, so, oh no, this one doesn't have the cartoon very well, but okay, if you see here, so this is the wheel. This is the NLR wheel itself. These guys, in the case of ZAR1, ZAR1 is detecting the pathogen effector indirectly through kinases known as RLCK, receptor-like cytoplasmic kinases. So this here are actually 2 kinases that are being detected by ZAR1 to be activated. So we think there's probably 2 possibilities. Most likely, the sensors are essentially functioning just like the kinases. They're on the outside of the wheel. They interact primarily through the loose-enriched repeat. And we also mapped the specificity of our network in the loose-enriched repeat domains. So we think this is the most likely case. Another possibility, which is actually known in the mammalian resistosomes, is they form heteroresistosomes, essentially, where the sensor would be one of the spikes of the wheel. But we think that's unlikely when you have these N-terminal extensions, because that will mess up this N-terminus, right? So we probably have both of these operating in different NLRs in plants, but certainly the ones where we have that N-terminal extension, we think that's most likely it's happening. It's decorating the wheel on the outside.
Sophien, very nice. Yeah. When you're looking at the N-terminal extensions and the sequence variant, it looked as though actually for monocots, there's a slightly different story, as though a lot of those, which are of the sensor type, are actually all in a single tribe. Is that right on your diagram? You've got things like
yes
NLA,
and no.
10 and...
Yeah. So...
is there something else that's happening?
Yeah, so, okay, I mean, where was that tree? It's coming later. Yeah, so I wouldn't say all of them. So in the monocots, indeed, there is, where is that? Was it?
2 or 3 back.
I went back.
In tribe 1.
Yeah, so, no, that's not the one. It was at the end, right? Yeah. So in the monocot, indeed, there is one major tribe that has a very ancient insertion, but that's not the only one. There's other insertions in other families and other tribes. In this case, also, it's an ancient one, because if you think about this clade here, which is well supported, so this entire clade, all of them have the N-terminal extension, right? So that N-terminal extension is a fairly ancient event, relatively ancient event. And that's the case, that's the published case in the monocot, is a case like that, where there was an ancient integration, and that has been maintained throughout quite a long time. But there are many, many of those examples throughout the NLR trees. That monocot case is not unique. And again, in a way, this is an example of that. I'm not sure if that's answering your question, if you had something else in mind.
Well, it's just that in that case, you'd actually have a situation where you aren't really looking at evolvability, because you've actually got both the mechanism and the recognition in the same molecule.
Yes, but these cases, in 2 cases, so both with PRF and SW5, this one, both of these cases, this N-terminal domain is involved in the pathogen effector detection. So this extra domain is involved in detecting the pathogen. In fact, at least 2 cases, this is virus resistance, that's bacterial resistance. Both of them, that domain is functional and is required for detecting the pathogen.
Okay, thanks for clarifying that.
Hello, Sophien. Thanks for the very nice talk.
Thank you.
I wonder, you show us that in tomato at least, these genes tend to cluster in the genome. I wonder whether members of the same cluster are more similar to each other than to other clusters in the genome, and whether that cluster in the genome is related to functionality.
Yeah, so the answer is simply yes and no. So there are both cases. So there are cases where there are clusters of phylogenetically related genes, which are probably tandem duplications, but there are also heterogeneous clusters. And one slide I skipped is exactly about that, because where is it? So yeah, this one. So in fact, this is exactly what this shows. So the original network and the one I told you about is this one. And I skipped this slide because I thought I wouldn't have enough time. But there's actually a subnetwork we discovered more recently, which we call the NRC6 network. And these are actually clustered in the tomato genome. So there's an NRC that is clustered with other sensors. And this is why we predicted these are probably working together. In fact, this is a nematode resistance gene called HERO that's been cloned from a wild tomato many years ago. And it's in a complex cluster in the tomato genome, which is quite variable between different tomato species. Interestingly enough, HERO could not be moved to potato, by the way. So it was not functional in potato. So now we're hypothesizing the reason it wasn't functional in potato because the NRC6 of potato is different from the tomato one. And you need to bring both players. But that's definitely a cluster that is conferring resistance. So it's kind of a sub-network from that big NRC network. So this is embedded still in that phylogenetic tree. Interestingly, the reason we ignored it initially is because we're looking at leaves only, and this is not expressed in leaves at all. It's root-specific. And then there's an RC0. And RC0 is a really interesting one. That's also cluster. And we call it an RC0 because this is the one that is ancestrally conserved in many of these species. And we think this is the one that's actually the progenitor of this network. And that one you still find even in tomato, amazingly enough. So even phylogenetically, they actually group together. And these genes are actually functioning together. We showed that. And they're also clustered in the tomato genome. So there's both situations, whereas this network is definitely unlinked. And there's players that are genetically unlinked that are working together.
But perhaps to readdress it in a different way, if you compare the sensor and the corresponding helper, are they usually more closely related than?
No, not at all. So that's the whole point. So back to this tree. No, the helpers are always in this clade, and the sensors are always in this clade, even with those clustered ones. So even NRC0, NRC6, NRC4, all those guys I talked about, they're all in this clade, and their sensors are always in this clade. So they originated from a pair, which was probably the NRC0 and a sensor. And then throughout evolution, they all diversified, but the helpers stayed helpers, and the sensors diversified and expanded as sensors. So that's why I'm talking about subnetworks that are embedded in this phylogenetic framework.
Rather, Joy?
So the redundancy that you're showing is really interesting from an evolutionary perspective because it makes the system, it enables the system to be very dynamical, right? And on the pathogen side, there's presence, absence, polymorphisms for the effectors, but then there's also some effectors that, like Talia showed, like in AVRE, and at least in a clade, are present. And those effectors provide an anchor that might be used for non-host resistance, or even if not, they're a target that the pathogen seems to not be able to escape. And so my question is, why isn't there a similar redundancy there?
Yeah, so redundancy on the effector side, you mean, or
for that subset of effectors that are core.
Yeah, it's not an easy. I think it's not an easy question to answer, and I think especially also going back to Talia's talk is that we are also not really studying the experimental system where this has evolved, which would be really looking at the wild potatoes in central Mexico, where Phytophthora infestans has, for example, co-evolved with this species. Why is there so much redundancy, and why is it that they're not relying on few effectors is a very, I don't know. I mean, I don't know. I don't have a simple answer to that, except to say that it's probably a very complex co-evolving system. But maybe I'll answer it slightly differently by telling you that, again, the work I didn't show you is that the pathogen effectors we have one from Phytophthora infestans, one from the nematode. They suppress NRC2 and 3, but they don't suppress NRC4, which in the phylogeny looks as the most derived of the NRCs, the most recent one. So it's possible to speculate that plants have this network, pathogen-evolved suppressors of these NRCs, which really take out of action a big chunk of our genes. And then the plant now evolved a variant of these NRCs to evade the pathogen suppression. But what's interesting is the pathogen doesn't lose that, right? So it still maintains that ancestral sort of effector, even though it's not completely effective anymore, because the plant found a trick to keep it. So usually these effectors just don't disappear, right? So it's a little bit like your trench warfare model, right? So they maintain, because if you lose them, then the pathogen is gone, because now on top of the new guy, you still have the other resistance. So I think it's a very dynamic system like that, right? And now maybe the pathogen will focus on mutating the effectors that are detected by the non-suppressed NRC. I'll make this very complicated, but I think I think that's what's going on. On top of that, if we go to Mexico, and this is a project I want to initiate, but haven't really had the time and to do it. If you go to the wild host of Phytophthora infestans, Solanum demissum is a classic wild plant, it's a wild potato in Mexico that's naturally infected by Phytophthora infestans. It's a hexaploid. And these genes, even in tomato, for example, they're present in multiple copies. So if you take all the copies of NRC2, 3, 4, for example, multiply that by 3, because they're usually present in 2 or 3 copies, 2 or 3 paralogs, then multiply that by 6, then you have a lot of potential to have actually new alleles that are kind of partially at least evading pathogen suppression and having maybe new network kind of affinities. So it's really complicated, and these systems just seem to have evolved by expanding the number of genes rather than focusing on the fewer systems. I don't know if it's a satisfactory answer, but yeah. Anyways, thanks. Good question.
Other questions? If not, I have one. So this is all about coil.
Yes,
NLRs. What about TIRs?
So first of all, there is no clear evidence of a network beyond pairs, but I suspect there are. And I suspect there are at least these kind of mini networks, or where I think, in my opinion, I think a lot of the pairs are actually like this guy or this guy, where they're actually mini networks that are in disguise, if you like. And people only study a pair of that, out of that. So I think there's at least that going on with the tiers. Whether they can accommodate forming genetically unlinked network or not, that's a really interesting question. And I also think that not all the CC-NLRs can form genetically unlinked networks, because some of these NLRs seem to function through negative regulation, which is not the case for our network, where there's one autoactive helper which is negatively regulated by its partner, its sensor. When the pathogen comes, that's relieved and then it's active. Those guys will be evolving as a genetic unit. You cannot uncouple them genetically, because those are death genes that cannot be basically segregating on their own. They have to stay linked with their partners. So I think there's an interesting aspect there, which is the evolutionary constraint determined by the mechanism of function. And that's something I think we need to really look at more carefully in the future.